All right. So how does geometry and topology can help tractography? I'll try to go quite slowly and give you intuitions about that. And I wanted to start with numbers that I think are maybe obvious for the OHBM community, but the brain is a very complex. It has more than 85 billion neurons. But a number that we rarely hear about is the amount of axons. So the amount of white matter, which can be more or less around 100,000 miles, so 160,000 kilometers of axons, interconnecting different brain regions together with short range connections and long range connections. So it's a real complex organ that we try to map with tractography at a certain scale, right? We have to keep in mind that tractography can be seen as the macroscopic scale, where things that we see are hundreds of thousands or several thousands of axons packed together in bundles or fascicles that I've colored here in red, yellow, orange, blue. And so all of these bundles have an associated cortex. And if there's an associated cortex, then you can associate functions. So it's really important to be able to map these connections and these tracks very precisely if we want to understand the brain and start doing proper connectomics analysis. And so one of the things that's possible nowadays is to use diffusion MRI, diffusion weighted imaging. There's been lots of technological advancements in the last, I would say, 20 years. Better and better images are coming out. Combined with anatomical images like T1 weighted images. I'll talk a bit more about that. We put the two together and we build a structural connectivity representation of the white matter. So this is often called the tractogram, the ensemble of all the tracks that represent the white matter wiring of the brain, which can be summarized into a connectome, the structural connectome. And now, a few years ago, we co-authored a very nice paper. So if you're interested in tractography and connectomics, I would definitely recommend reading this paper about the challenges of mapping the human connectome with tractography with current technology. So it's not a solved problem. It's a very difficult, ill-posed problem. And I want to take the next 20 minutes to maybe highlight what geometry and topology can help us to try to improve tractography and improve mapping of the connectome. So I don't want to focus on just have a depressing message today. I want to focus on problems, but also solutions to these problems that have appeared in recent years. Why is it so hard? A key reason for it is because it's full of crossing fibers everywhere. So you have to imagine these 100,000 miles of axons packed into a brain. And so there are numerous regions of crossing fibers. So two-way crossing, three-way crossing. And so if we have to pack all this connectivity within our small head, well, it's estimated that you can have up to 90% of imaging voxels that currently have crossing fibers. So you can imagine for a tractography algorithm, it's a nightmare to traverse. It's very easy to get lost and very easy to reconstruct non-existing anatomy. And so out of this paper, one of the key citation or message from the paper says we need a revolution in tractography. We need better ways to fully reconstruct the existing anatomy, so the real true positives, while controlling for all the false positives. So all the mistakes and all the wrong turns that our tractography algorithm can make through this maze of local orientations in MRI data. And just to highlight this, you see on this graph, this is a bundle overlap over overreach. And you can see that out of the hundreds of algorithms that were tested in this paper, most algorithms barely fill up 50% of the white matter of the existing anatomy. So a lot of the white matter is just not explored. And so, yeah, around, between 40 to 50, 60%, depending on the algorithm and depending on how aggressive this algorithm is. And the more aggressive the algorithm is, the more mistakes it makes, of course. So it's this specificity and sensitivity trade-off of our current tractography technologies. And just to show you a few bundles, it's been quite established that some bundles are harder to track than others. So the blue bundles on the top right corner are the easier to track bundles. We call them medium difficulty. The middle pack is hard to track, and we have very, very hard to track bundles that are very small, that are connected to very small part of the anatomy. And so just to highlight that this really points to the fact that tractography is not as good everywhere in the brain. Some regions of the brain are harder to track than others. Why is that? And here's just a brief list. And this is what I'm going to focus on today. But it turns out that the very deep white matter with the big highways of the brain are easier to track. But near the cortex, near the basal ganglia, near the brainstem, the cerebellum, these locations of the brain, which is linked to topology and to geometry, because of a numerous number of factors, are very hard to track. And so the consequence is that they're underrepresented in our connectomes. So when you extract the connectome nowadays, you have to keep that in mind. Some tracks are less represented, not because they're less connected or because they're not as healthy, but just because they're harder to track, so they're harder to reconstruct. So let me focus maybe on this problem first. Are there ways to increase specific sensitivity? Can we track more? And so this is where topology comes into play. And this is my chance to salute Giorgio Innocenti. Giorgio was a mentor to me and a colleague when I was in a sabbatical in Lausanne. And unfortunately, Giorgio passed away while skiing in the Swiss Alps during the pandemics. And so I raised my hat to Giorgio, and we're very sad of losing him. But basically, Giorgio is a guy that's been tracking individual axons since the beginning of his career. So he knows how topology and how different signals lead to axon going from a source to a certain target. And Giorgio is the guy that was telling me, you just cannot let tractography do anything you want. You have to guide it. We have to use cues from topology and from anatomy to improve tractography. And so this is a very nice read if you want to see how ideas from axon tracing can be kind of transported into the macroscopic scale diffusion tractography. But basically, what is clear is that orientations are not enough. What you see here is a zoom into a crossing fiber regions where the corpus callosum crosses the blue track, the motor track, the green track coming out at you is the language track. And so if you're just an algorithm stepping into this field of very complex crossing structure, you'll get lost. Not only you'll get lost, but also it's very hard to reconstruct. Here you see the blue track, it should be full. This should be connected to the whole motor cortex, the whole motor homunculus. But tractography is attracted by the easy tracks. It's attracted to the centroid of the easy to track regions. Same with the corpus callosum here. So a key conclusion, I think, in the tractography community these days is that local orientations are not enough. We need more than just the diffusion signal. We need extra priors. We need more anatomy. We need more modality. And so this is illustrated here very well, right? When three fascicles cross here between the red, blue, green bundles, it creates a blockage for tractography. It's a huge traffic jam. And if you're a tractography algorithm trying to traverse this traffic jam, well, either you get lost or it's very hard to traverse, even on the best existing data. Actually, the higher the resolution, the more difficult it is to traverse this crossing fiber region. So we need some more information. We need to inject somehow that as you're tracking from down to up or down to up to down, you're not allowed to do and jump onto the green track. If you're on the blue track, you should remain on the blue track. And so this is the notion of axon development and prior knowledge from anatomy that we try to inject in this paper. So just to, I don't want to, spend too much time introducing it, but just to promote the idea that if you know what you're looking for, it's possible to better reconstruct it by better priors about where you start, where you finish, and what are the actual orientations that should be legal to track. So this is extra priors to help tractography fill up the existing anatomy better. So, and get more of the hard to track regions. So for instance, we've applied this to the fornix because in Alzheimer's disease, the fornix is very difficult to track. It traverses a lot of ventricles and partial volume. It's very highly curved. And so we need strategies such as bundle-specific tractography to do a better job. Back to our list of problems, I want to talk about the cortex next, because I believe this is linked a lot to connectomics, to fMRI, and this is a very hard region to reconstruct well at the moment with tractography. And here's the problem. Under the gyrus or the gyral blades, tractography doesn't have a lot of resolution and tends to go straight up into the crown of the gyrus. We don't see the very nice fanning structure, and all the gyral blades and even the sulcus are not well connected by tractography. And I'm going to show you some numbers later, but you'd be surprised to see that tractography barely reaches 50% of the cortex, right? Which is a problem if you're thinking about structure-function relationship, relationship with EEG, MEG, fMRI. We need better tractography that really covers the whole cortex, or as much as possible of the cortex. And so this has been called the gyral bias by Van Essen. And a lot of the AHCP, Human Connectome Project Consortium, I've tried to come up with tools to better assess this gyral bias problem. And this is where geometry comes in. This is where, if you know the shape of the cortex and you have a surface representation of the cortex, well, then you have information about the normal. You have information of where is the cortex have positive or negative curvature. And you can help tractography better start and better end. And so here's an illustration. In blue, here are the negative curvature, and in red, the positive curvature, and in green, kind of the zero curvature where you have the walls. So you can see that if you have a nice T1 segmentation of the cortex, it's kind of easy nowadays to get geometrical features on this cortex. So you can have it at the gray matter, you can have it at the white matter, gray matter interface. And so our objective in the last 5-6 years with Etienne Saint-Onge in my lab was to try to bring this into the tractography world. And here, notions of computer vision came into play. So we implemented a mass stiffness geometric flow that kind of allowed the cortex to unfold itself, to try to see how can we let the cortex unfold itself and connect with deep white matter where we have better signal, better orientation, and better diffusion data. And so if you do that, if you track the process of the cortex unfolding itself, this is without any tractography, without any diffusion data. You just take your cortical mesh and you let it deflate. Well, you can see that you reconstruct kind of the orthogonal fanning structure under the cortex. And so this is at the heart of surface enhanced tractography. this geometrical flow will help seed and terminate tractography in a new computational framework that we called SET, Surface Enhanced Tractography. Basically, SET can be plugged in any underneath existing tractography algorithm, whether it's MR tricks or deterministic or probabilistic tractography. It's a layer on top of it to say, how do we start and how do we end tractography? And here's a nice illustration. You start from the gray matter, white matter interface. You let the geometric flow inwards. This traces trajectories. So this will help see tractography in the right directions to go towards the deep white matter. But not only that, as tractography hits this surface, we now have a model of where it came from. So we can project it back to the cortex. So it's kind of the glue between deep white matter, superficial white matter, and white matter, gray matter interface. And thanks to that, we can, I'll show you some results, but we can get much nicer reconstruction of tracks that penetrate the cortex, that connect not only the crown of the gyrus, but also the blade of the gyrus. And it's much less noisy than the existing tractography algorithms. But still there, using the best technology that we could do in this 2018 paper, even when I use SET and surface enhanced tractography, you can see on the right here, I still have 30% of the cortex that is not connected. Wherever you see dark gray or black area, these are mesh points and triangles on the cortex where a track does not reach the cortex. So you can see this can really bias our connectivity analysis. Only the crowns and the very easy to track regions are connected, even with our latest geometrical approach. And this is another illustration here, but here the hard to track region, the empty regions of the cortex are in purple. So can we do better? Can we push this even further to try to maximize the number of tracks that connect to the cortex? Well, yes, this is our latest paper off the press, again from Saint-Onge. So this is all part of his PhD thesis. Now he's a postdoc fellow at McGill University. But the idea is to use adaptive and dynamic seeding to go even further. Now I'm going to zoom in on the cortex. The cortex comes with very rich information. I already said about the normal and the shape, the positive versus negative curvature. But now you could imagine that we can bring any information in the process, such as cortical thickness, volume, labels from an atlas, the local density of the cortex, etc., fMRI bold correlation, PET. So I'll show you some ideas behind. None of this is done here, but now the computation framework is set to do such thing. So now the options is you can do either you seed everywhere the same, or you can impose some priors on what you're looking for. Here in B, you can see cortical thickness. So the more red, the less, I think, or more, yeah, the more thick the cortex. So you can decide to start more seeds for tractography. You can decide to just look at a sub part of the network and the cortex. You can use cortical volume. You could eventually use fMRI or any other signal because, as I said before, the mesh or the graph here, the surface is the link between all these modalities. And the other thing we implemented is a dynamic seeding approach. So we're going to have now a new strategies to do tractography in a dynamic iterative way. So if after a first tractography algorithm, you get the image in 2, and you see that you still have a lot of places where the cortex is empty, you're going to take the difference between your original seeding map and your termination map of all the tracks where they terminated, and you're gonna keep seeding. So you can see in 3, you're gonna seed basically from the sulcus and the walls of the cortex, and you're gonna barely not seed anymore from the crown of the gyrus, to try to push and push and push and push tractography to try to find hard to track regions and connect it with proper anatomy. So of course, this is very new. This has to be validated, evaluated. But so far, based on our test-retest database of the Human Connectome project, this really seems to improve connectivity and help. So here is a first image. If I just use classic tractography with intersection with the voxel-based free surfer map. So white matter, gray matter interface from free surfer, and I just intersect tractography with that, just like most standard connectomics pipeline. Well, you can see on the left here that the occipital cortex, the motor, precentral and postcentral cortex are overrepresented with tracks. These are the easy to track regions. And on the right, you see where the connectome is most variable. This is the coefficient of variation, average for all test-retest pairs. Here we have 45 test-retest subjects. So we can really do robust statistics here. And so you can see that all the yellow spots on the cortex are very variable. So some regions of the cortex are well tracked and have little variability, and other regions of the cortex have very high variability and are very poorly represented by tracks. So if we use set and dynamic set, we can really start better distributing endpoints of tractography. So we increase cortical coverage. Now almost 99% of the cortex is covered by tracks. So that's the first good news. Second good news is we reduce the gyral bias. That is the crown, the wall, the sulcus now have connections underneath. And we improve the variability. So the variability across the cortex here, you can see, is very blue, which is reassuring, which we think will help us have better structural connectivity studies and maybe better connectivity studies that are joined with fMRI. So all that to say that this truly seems to improve connectivity. And although this is very recent, we have tools in Python to do this and to help researchers apply this in their connectomics pipeline. And so please contact us if you need any help. I'd be happy to put you in touch with Etienne. And so nowadays our pipeline looks more something like this. So from raw diffusion data and a good anatomical T1 image, yes, we still have our good local fiber crossings, but we include surface enhanced tractography and maybe in the future dynamic and adaptive tractography to get a better connectome, more coverage of the cortex. We combine this with a cortical parcellation, and we can build a connectome that is better than the standard tractography, thanks to a few geometrical and topological rules. And so I think this is the perfect computational framework to integrate more stuff into this in the future. This is the perfect computational backbone for multimodality. So that's what I call here, but we're really going towards multimodality connectomics, where we want to bring fMRI, PET, metabolic component elastography, microstructure from myelin MRI or from diffusion MRI. And I have a vision that the cortex and geometry could help fuse all these information together. I want to leave you with a thought. Keep that in mind. Of course, everything we've developed here is based on very nice, high quality human connectome project data. As we go to the pathology, as we go to brains with lesions, with atrophy, such as an Alzheimer's brain here, this may very well break and challenge some of our techniques that assume a certain geometry and a certain topology. And so we will for sure have to consider this in the future years. With that, thanks again, Mo, for inviting me. and happy to answer some questions.
Okay, thank you for this amazing talk. Anyone has any question to the speaker? I have a question. So you said that there are up to, you shown that the 2 crossing fibers and 3 crossing fibers. Is three crossing fiber the maximum number, or can you have a more crossing?
Yeah, I think that's an open question. Most often we limit our crossing fibers to 3.
I see.
But I think if you allow yourself to have very tight angle crossings, then it's an open question as to know, is it branching structure, fanning structure, or pure crossings at a tight angle? So you could imagine having maybe 2 crossings in the plane and then 3rd and 4th crossing in the other axis. So you could see locations in the brain with 4 and 5, but it's hard to know if it's a single peak fanning or a single peak with a tight crossing. So it's a bit technical, but this is still an open question in the world of diffusion MRI.
So this crossing fiber problem seems like a deep learning problem, is it? Like if you apply some sort of deep learning, can you get a much better?
Yes, yes.
So anyone actually done this, or?
There are a few papers out there trying to do that. Yes.
I see. Okay.
And of course, if you know a little bit about DTI is a single fiber model. So it averages everything together in the voxel. So keep that in mind when you have FA in a voxel. It's actually the contribution of every fascicle going through the voxel average into a single value. So we're trying to get towards more fiber-specific FA or fiber-specific myelin content or axon content. So there's actually a new term called the pixel for the fiber element. So you have the pixel, the voxel, and now the pixel for the fiber element in each voxel.